Good afternoon, everyone, and welcome to the Van Andel Institute public lecture series, From Fish to Dish, How a Little Fish Can Help Cure Cancer. My name is Steve Ozinga, one of the philanthropy directors here at Van Andel Institute, and I'm glad you can join us today. Today, we'll hear from Dr. Stephanie Grainger discussing and answering the question, can a tiny fish frequently found in kids' aquariums help us cure cancer? Stephanie will share how zebrafish help scientists better understand cancer and fuel new insights that one day could lead to powerful new treatments. A little background on Dr. Grainger. Dr. Stephanie Grainger is an authority on the biological underpinnings of how stem cells develop and how these processes can go awry during cancer. She earned her PhD in cellular and molecular medicine from the University of Ottawa, where she investigated the role of CDX transcription factors in intestinal development. She then completed a postdoctoral fellowship at the University of California, San Diego. Her research during that time sought to understand the role of Wnt signaling in blood stem cell development, work that she continued as an assistant professor at San Diego State University. During this time, she discovered a novel stage of blood stem cell development, which is driven by a specific Wnt cue. Furthermore, she discovered a novel mechanism of Wnt signal reduction or regulation, In 2021, Dr. Grainger joined Van Andel Institute's Department of Cell Biology. So if you find yourself with questions during the presentation, please submit them via the chat function. We'll have a formal Q&A session when Dr. Grainger is done speaking. So let's begin today's program. Please join me in welcoming Dr. Stephanie Grainger.
Thanks, Steve. Hello, everyone, and thank you for coming out today. So my name is Stephanie Grainger, and I'm an assistant professor in the Department of Cell Biology here at the Van Andel Institute. And I'm excited to tell you folks a little bit today about how we can use zebrafish to help make cures for diseases like cancer. So in the lab, we study stem cells. And one question that I get a lot is, what exactly is a stem cell? And so every tissue in your body has stem cells that replenish that tissue. They're the building blocks that make your entire, for example, skin or intestine or heart, or in the case of what we study for the most part, blood. So what you're looking at here is a diagram where we have a stem cell on the left, and it has two main jobs in life. The first is to differentiate into mature cells, and the second is to make more of itself or to self-renew. And so we're interested in these hematopoietic stem cells, or HSCs, or you can also think of them as blood stem cells. And these are the cells that are restricted to the blood lineage, and they make all of the different types of cells in the blood. So you've probably heard of red blood cells and white blood cells, and so that's what we're looking at here. If we get a disease of the blood like leukemia or lymphoma, one or more of these progenitor cells acquires a mutation that gives them a proliferative advantage. So what that means is that they make more and more of themselves until eventually you actually don't have space in your blood anymore to make these downstream differentiated cells, and we just have too many of these progenitor cells. And the way that we treat leukemia and lymphoma right now is to irradiate a patient's supply of diseased blood cells and to give them back a healthy long-term HSC in the form of a bone marrow transplant. Now that actually works quite well. And you might even know someone who's had leukemia or lymphoma who has had a bone marrow transplant and is doing very well right now. The problem with this is that we actually don't have enough donors that are matched to the number of patients that need a bone marrow transplant or could use a bone marrow transplant. And so if we could figure out instead how to make these HSCs in the dish, we would be able to circumvent the need for a bone marrow donation and have just tons of these blood stem cells available for doctors to administer when needed. And so this actually isn't a new idea in the field. This is something that people have been thinking about for about 40 years now. And the idea here would be that we would take a skin cell, for example, from a patient, and we would reprogram it into a naive cell that we call an induced pluripotent stem cell. Then we could differentiate those induced pluripotent stem cells, or iPSCs, into specific cells that match the patient's genetic profile and give them back HSCs that have been derived in the dish to match them. And so if we could figure out how HSCs are made normally in an animal organism, for example, we'd be able to take the genetics that animals use to make HSCs and apply those to the dish to be able to make HSCs for humans. So we use a critter in the lab called a zebrafish. Some of you might recognize this from the pet store, actually. You might know them more commonly as zebra danio. And so one of the main advantages of zebrafish you're looking at right here. This is a male and a female fish that are mating. And along the bottom of the tank, you can probably appreciate that there are these tiny little whitish orbs. Each of those orbs is a fertilized embryo from these zebrafish. So you can see that from a single mating pair of fish, we can make about 100 different embryos to look at. And also you can probably appreciate that these fish are laying the eggs on the outside of their body, in contrast to other organisms like, let's say, mice that develop their babies in utero or in the mommy's tummy. And so one of the advantages of that is that we can actually watch development as it's happening. So that's what you're watching here. This is a video from about 0 to 24 hours post-fertilization. On the outside, you might see a grayish sort of circle. That's an eggshell that protects the embryo as it's growing for the first few days. In the middle, there's a big blob, and that's the yolk that feeds the embryo. And you'll notice that there's sort of a crescent moon backward C shape that's moving around in there, and that's the embryo itself that's developing. In a minute, at about 2 o'clock, you'll see the eye pop up from the embryo, and the tail will pop up around 4:30. And you'll also see in a few seconds that the embryo will start twitching, and that's because his muscle fibers have started developing. That happens after about 24 hours of development, which is the same time that fish start to make their blood stem cells. This is much faster than it happens in mice, so it's pretty useful for us because we can do our science a lot faster and make more impacts. You can also appreciate here that these fish are see-through, so we can actually label different cell populations inside of the embryo and watch them as they're being born. So what we're looking at here is a zebrafish in the cartoon here that is about 2 days old. On the left, you can see that's the eye of the fish. Labeled in pink and peach are the major blood vessels in the fish. And I've pointed out the aorta, which is the site of where blood stem cells are born. Once they're born there, they travel to the caudal hematopoietic tissue, which is pointed out over there in the tail. And you can actually appreciate here from this video that's taking a zoomed in image of part of the tail and part of the aorta there, that we can actually see those blood cells as they're circulating around in the fish. So you can imagine that we can do some pretty cool studies here where we change the genetics of the fish and we ask how that impacts on their blood development. Here's another video here just to orient you. On the left-hand side, that's where the head of the fish is, and it's pointed in towards the screen. The future spine is facing towards us along the horizontal axis. And so what you're watching here is a time-lapse video where we have blood vessels in green. So these are the cells that will go on to form blood vessels and also blood stem cells. And we have these cells called somites, which are a transient block of cells that at this stage of development are going to instruct nearby tissues on what they want to be when they grow up. So what I'd like you to appreciate from this video is that those green cells start on the outside of the somites and they crawl in underneath the somites. As they're crawling in underneath, the somites are telling them, you should be an endothelial cell when you grow up, form part of the artery. And they're telling other cells, you should be a blood stem cell when you grow up. So this stage of development, as those somites are telling those endothelial cells who they should be later on, we call that specification. This is when inductive cues instruct future HSCs that they should become HSCs. After specification, when the fish looks a little bit more like this, around 26 hours post-fertilization, we can start seeing those cells that got told they should be HSCs undergoing a process that we call emergence. So what happens is they start out as an endothelial cell on the bottom of the aorta, and they actually change their identity into a blood cell. So they bud out of the aorta, which is a stationary tissue, and they become a circulating blood cell. And those are the blood stem cells that feed our entire blood system for the rest of our life. We can actually watch that happen in real time. So To orient you here, we're looking at a really, really zoomed in image of the zebrafish side on. So the blood vessels are labeled in red, and you can think of the blood vessels sort of as two plumbing tubes sitting one on top of the other, running from left to right across the screen. And if we took a section, so we just chopped right through those tubes, we would see on the top the aorta and on the bottom the vein. And the blood cells themselves are going to be labeled in green in this video. So what I'd like you to draw your attention to is underneath this bracket in the line that's running horizontally across the screen, that's the floor of the aorta. And I hope that you can appreciate that these cells start out red as endothelial cells, and they actually start changing their color to green and moving around, and then they'll actually exit the aorta and they'll start circulating through the vein. So what you're watching right now is the birth of a blood stem cell, which is something that we can do in zebrafish that you can't do in other organisms. So the way that cells decide who they should be when they grow up, or what they should be doing with their lives, or where they should be going in the future, is by talking to each other. So cells can talk to each other using proteins such as the Wnt pathway. So what I've illustrated here on the top in this normal circumstance is that there's a stem cell that differentiates into a mature cell, and it also makes more of itself in a process called self-renewal. If we don't have enough Wnt happening in our bodies, what happens is that those stem cells stop self-renewing and they differentiate towards mature cells, and then we lose that stem cell pool. And when we have too much Wnt, you can see what's happening there is that we block that differentiation, and these stem cells keep replicating instead. So you can imagine that if we have way too many stem cells, they start making way too many more of themselves. And that sounds a lot like something you might understand as cancer. So one of the questions that we asked in my lab is, how does Wnt signaling impact on HSC development? So to get at that, we used those zebrafish that I just showed you the video of, where we saw those little green HSCs emerging from the floor of the aorta. And we treated those fish with compounds to either turn on or turn off Wnt signaling during development. And then we counted the number of blood stem cells that formed in response to that. So as you can see here, we're looking at that same really zoomed in image that I showed you the video of, but in this case, we're looking at 3 different conditions. On the top, we have the control. So these fish weren't treated with anything. This is just how they normally develop. In the middle, we've taken away their Wnt signal. And on the bottom, we've added some more Wnt signal to them. And so what I hope you can appreciate is that if we compare how many of these little arrowheads are counting the different HSCs, when we lose Wnt signaling, we lose HSCs. And when we gain Wnt signaling, we gain HSCs. So like in other stem cell populations, Wnt control the number of HSCs that are formed. And so the next question we wanted to ask was, is Wnt important during this specification time window when those endothelial cells are receiving those inductive cues? Or is it actually more important during that phase of emergence when those cells are budding out of the aorta? And so to answer that question, we turned Wnt on or off during these time points of specification and emergence and asked how that affected the HSCs. So what you're looking at here is a control fish. This is a slightly different readout of HSCs. So rather than those red and green cells that show up as yellow being the HSCs, what we're looking at here are these blue-purple cells are HSCs. And if we take away Wnt, I think you can appreciate that there's fewer of those HSCs. And the first diagram there, we're taking away Wnt during the specification window. If we take away the Wnt during the emergence window, however, you can see that there's the same number of HSCs. And when we do these types of experiments, we count a lot of different animals to make sure that this is a consistent process that happens. So on the right, you're looking at a graph where we've just counted a lot of different HSCs in a lot of different animals. And so what we conclude from this is that Wnt is needed during that first window of HSC specification when those endothelial cells are crawling in underneath the somites. So one might ask, how does this really impact on human cells? So this is a zebrafish that you might find in your aquarium. And do these things really recapitulate themselves if we look at human cells? So we have a system in the lab where we can look at the specification and emergence windows of human cells in the dish. So what we can do is add different compounds to those induced pluripotent stem cells, and we can push the cells towards a blood vessel and then also towards blood stem cells, and we can count the number of cells that form. So what we did was we either activated or inhibited Wnt signaling during either that specification or emergence window, and we counted the number of cells that form, and you can see the cells, what that readout looks like for us here, where the blood vessels are in red and the blood cells are in green. And so when we did that experiment and we inhibited Wnt signaling during the specification window or activated it during the specification window and counted the number of cells that form, you can see in this graph here on the y-axis, we have the number of blood stem cells that form. So the higher the bar, the more stem cells there are. And along the x-axis or the horizontal axis, you can see how many conditions that we've looked at. So in the untreated condition, we get about 12 of these cells. And when we add Wnt, we get about 22. And when we take Wnt away, we get more like 6. So what this tells us is that Wnt is also required during this specification window in human cells, and it has the same impact as it does in fish. And we also did the experiment where we took Wnt away or added Wnt during that emergence window, and you can see that all those bars look the same. So what that means is that there was no impact of Wnt signaling on the emergence window. So altogether today, what we've talked about is that zebrafish make Wnt happen. Zebrafish are a good model for looking at how blood stem cells develop. And we looked at two different important time windows. So we looked at the specification window when those endothelial cells are crawling in underneath the somites and receiving inductive cues telling them who they should be when they grow up. And then there's this emergence window when those stem cells start blooding out of the endothelium and circulating through the blood system. We can recapitulate that kind of a system in a tissue culture dish using human cells, and we have a specification and emergence window just the same. And I also told you that Wnt is required during this early specification window and not during this emergence window, and that drives between humans and also zebrafish. And so I just wanted to emphasize that this type of work takes a lot of people to do. So this is actually just a subset of the people that have been involved in these kinds of studies. At the Van Andel Institute, my lab here has 8 people in it. And so you can see that these people are all, except for Kelsey, we don't have a picture of her yet. These people are all involved in different aspects of how we progress with our science. Before I was at the Van Andel Institute, I was at San Diego State University and the University of California in San Diego. And you can just get an idea of how many people are involved in these types of studies. Also, we have a lot of excellent support here at the Van Andel Institute from our operations teams and core facilities that help us with the logistics of actually doing science. So everything from, sending and receiving packages to helping us image things on microscopes. And we also have a variety of collaborators that help us out with different aspects of our scientific endeavors as well. And lastly, we also receive funding from various generous donors and foundations and through grant writing to government agencies. And so with that, I would be happy to take any questions.
Great. Stephanie, thank you so much for that fascinating presentation on zebrafish. If you have any questions, those listening in the audience, please type them in the chat room and we will get to them. A question to start that we have, Stephanie, why zebrafish? How did you get started with this particular fish, and how long have you been working with zebrafish?
It's a great question. So when I did my PhD, I used a mouse system instead of zebrafish. And one thing that really frustrated me with that was that we couldn't actually watch these processes happen. And so I was really drawn into working with zebrafish because they're translucent and we can watch these developmental processes happen in real time. And I've been working with them for about 10 years now.
Wow. What's the coolest thing you've found out about zebrafish so far?
That's an interesting question as well. So I think one of the coolest things that we can do with these fish is watch blood stem cells being born. So this process of labeling different cell populations and seeing them just arise out of nowhere and watching where they go and watching the cells that they interact with throughout their lifetime, that's a really cool thing for me.
Good, fantastic. Your particular research in your particular lab, what are some of the things that you are all very excited about in the future of research? You know, what you particularly do with the zebrafish and such?
I think what we're most excited about is being able to potentially translate some of these findings into human cells and getting towards human treatments. So the implications for our studies and how we are able to make blood stem cells in the dish in the future and whether or not that will actually lead to cancer cures, I think that's a really salient feature of working in the lab.
Fantastic. Are there, and this is kind of a piggyback question on that, are there any gaps in knowledge that you hope to find, given what you know so far? Is there like a missing area that you really want to focus on?
Yeah, so we study how Wnt signaling affects these HSCs, and I kind of told you the beginning of the story here where we know it's required during that specification window. But what's really interesting is that there's not just one Wnt gene encoding in our bodies. We actually have 19 different Wnts. And so what's really interesting to me is trying to figure out which of these Wnts is actually needed for making blood stem cells, and if different Wnts have different effects on blood stem cells so that we can translate our findings to use the right Wnt when we actually do make them in the dish.
Fantastic. Thank you. A couple questions have also come through from the audience. Sam wants to know, what hope do you have in impacting bone marrow disease, and are we collaborating or working with other institutions in our work?
Yeah, so we don't have any formal collaborations just yet with, say, clinicians to be able to translate these findings because it's a little bit early, but there are labs working around the world on how HSCs are made in the dish and working towards getting that therapeutic HSC that will be able to transplant into humans.
Fantastic. Dee has a great question. Any idea how long before human testing will begin?
That's a really great question. I wish I knew the answer. That's why scientists at the Van Andel Institute are coming in every day and working hard on these kinds of things because we really don't know. And hopefully it's soon. Okay,
fantastic. That's the end of the formal questions. I don't see any more in the chat room. Personal question to you, Stephanie. When did you know you wanted to be a researcher or scientist? When did that spark, in your mind light up? What age and how did that process work for you?
Yeah, so when I was growing up, my mom was a family doctor. And so I'd always been interested in having an impact on human health and disease. And so I went into an undergraduate program in biochemistry. And somewhere along the way, someone suggested to me working in a lab as a good experience. And I kind of got addicted to doing science during that first lab experience. I just find there's something really thrilling about looking at data that nobody else in the world knows about. It's like this great secret about biology and how it happens.
Fantastic. Good. No, thank you. And I think that's, don't see any other questions. One just came in, but it was similar to what we just talked about what might have led you to the specific field of research, those types of things.
Yeah.
Any other thoughts, Stephanie, before we wrap up here?
Thank you for giving me the opportunity to teach everyone a little bit about what we do in the lab. And I'm glad to have been able to do that for you today.
Fantastic. Thank you, Stephanie. And thank you, everybody online, for joining us today. I also want to thank Dr. Stephanie Grainger once again for her presentation and insight. I invite you to stay active and engaged with the amazing work happening here at Van Andel Institute. Our work continues to provide hope for those impacted by cancer, Parkinson's, Alzheimer's, and many other diseases as well. Please visit our website at vai.org to keep up to date with Van Andel Institute events, and whether virtually or in person, we hope to see all of you at a future event. And thank you once again for attending today, and have a great rest of your day. Thank you.